And yeah, so to start, as per usual, did anybody have questions about lab 2 that they want to go over, or the pre-lab from lab 3? I just graded those earlier today. I haven't entered them yet, but seems like people were doing pretty well on those. But if you have any specific questions, I can pull that up. Feel free to throw it in the chat or just speak. And then also remember, if you don't have any questions about lab 2, that's great. But if you do, I have my office hours tomorrow morning from 9 to 10, usual link, usual time. And then also remember that your post lab 2 is due tomorrow by the end of the day. So we had, I think pretty much everyone got theirs in on time, but just remember by midnight tomorrow, if you can, or yeah. So just turn that in as soon as it's done, so you don't have to worry about it at 11:55 or something. I got a question about if it's possible to earn points back on a post lab. So no, but I believe your lowest lab score is dropped. So if you're worried about one particular one that is just more difficult than the others, like don't worry, that one will be dropped. I think it's like the whole lab as a cluster, but I'll check with Pat about that. But yeah, if you're, and also like if there's certain questions that you're confused about on the post lab, you're always welcome to come to office hours and we can talk through them. I can't tell you the answer, but we can help work through it and make sure that you're understanding the question correctly. So yeah, it's always good to kind of do that stuff as early as possible. So yeah, and so everyone should have read through the lab. And so hopefully kind of get a sense of what you're going to be asked to do. And so we can maybe go over some of those questions that you have today also. Okay, so no other questions about what we've done so far. Awesome. Is my PowerPoint still sharing? Yes? Okay, cool. I don't see the green box around it, but that's good. Okay, so for today, jumping into our activity for the day, we're going to be talking about population growth in 2 acts. So the first part will be estimating population size. So these different methods that scientists can use for estimating population size, because as we know, and as you know, because you've all sampled your habitats, it's kind of hard to A, find all of the species, and then B, find all of the individuals from every species. But still, biologists want to have some idea of how many different individuals there might be of a species. This could be, you know, if you have a species that's endangered and you want to get an accurate count of how many there are left, or if there's potentially an invasive species and you want to know how many you're dealing with. So this is a really important aspect of biology, but you can't, oftentimes it's impossible to count every single individual. So there's lots of different methods for kind of approximating that and getting some idea without having to count everything. And then kind of the second part of the lab is we're going to both think about population sampling and estimating population size in the context of this kind of bigger building a research question and forming a hypothesis and kind of establishing methods. So this is the process that every researcher goes through of you have to form a question, a hypothesis, how you're going to sample that. And then for this lab, we're going to be looking at that through the lens of population sampling. So it's kind of a good way for these two things to go hand in hand. And then remember the homework, as per usual, is a Word document or PDF uploaded to Canvas, responding to the questions. And then there are 2 videos for part 2. They're like a video of the blepharisma, little one-celled dudes moving around, which is super cute. And I think in the normal lab, you'd be able to actually look at them. But this is the next best thing, because in the real lab, you're still looking at them through the microscope. So, and sometimes they can be hard to find. So this way it's just easy, you can see them. And I think there's a practice in there of like being able to see them on the screen. So it's just great all around for learning how to identify these little guys. And then there's also video for the data collections for questions 4 through 6 on the post lab. And these links are in your lab document as well. So kind of a general overview of the lab topic of population growth. So something to think about as we're moving through this, or as you're thinking about these questions, is what is a population? So does anybody have any ideas about how they would define a population? You could do it privately if you're not sure, but just, you know, throw out some things. Group of individuals we want to know about. Anyone else?
It's a group of individuals that live in certain habitat.
Yeah. Number of a particular species, totally. Yeah, these are all like totally getting onto the right definition of, it's not everything in a species necessarily. It's a population is kind of, could be distinct or nebulous or whatever, but it's like a group of individuals of a certain species of interest. And so there are some populations that are really isolated, and it's very easy to identify that they're a part of one population. Sometimes there are these things called metapopulations, that these populations can kind of move around and go into different groups, and they're really connected. So it's totally makes sense that we'd have kind of different definitions of population, because population can be hard to define. But yeah, you are totally right on the right track. It's just important to note it's not the same thing as a species. It's like a group within a species. So, and then another thing to think about is what is the difference between exponential growth and logistic growth. So I have kind of two curves of exponential and logistic, and I believe this has been introduced in lecture, right? Logistic and exponential. Sorry, let me be able to see everyone. One second. Okay, now I can see everybody. So this has been introduced in lecture. Okay, awesome. Yeah, so it's these two curves, and then basically the difference here is, oh, sorry, I was gonna ask. Can anybody tell me which one is the logistic curve, on the right or the left? Yes, excellent. Okay, great. So then by definition, that was the logistic, the other one's the exponential curve. And so the key here is that carrying capacity, right? And you talked about this in the pre-lab. So the concept of a carrying capacity should be totally familiar to you. Does anybody remember what do we mean by density dependent and density independent growth? This one's a little tougher. So if there was density dependent growth, would it be hard, would growth be constrained if there were more individuals in a space? Yeah. So exactly that. If there's density dependent growth, the more individuals in a space, say there's a carrying capacity or something, or they're in some, I think they had the graph with the penguins in there, and they talked about like, okay, if there's density dependent growth and there's 1 million penguins in this tiny spot, then it would, each additional penguin would make it harder for the penguins to continue growing their population. Their resources are more constrained. Potentially there's not enough resources to reproduce, so deaths might be higher, births might be lower, so density dependent. And then there's also situations where there's density independent growth, where the density of the species doesn't seem to affect their growth rate. And then this was the final question on the post lab, but what kind of factors can influence the growth of a or this was kind of touched on when they were talking about density dependent growth, but what factors can influence the growth of a population? So kind of just throw out a few things that you learned about that you think could affect the growth of a population. Could be intrinsic or extrinsic. Yeah, so we've got abiotic factors. So a disturbance in the area could totally affect the amount of resources. This could affect the growth. It could also like deplete the number of reproducing individuals. So that's a good example. Then they also talked about the carrying capacity. If there's a K value for a certain area in a certain population, that this could affect the growth rate. Because remember, as you can see in the logistic curve, kind of slows down as it gets towards that carrying capacity. The R value, so the growth rate of the species could affect it. And that's maybe more of an intrinsic factor. So lots of different examples for what could affect the growth rate of a population. And remember that it can come both from the population itself and then also from the environment. So we were talking about, disturbance, fires, habitat destruction, all that stuff. Any questions about population growth, the material we've covered so far? Great. All right, so that was kind of a review of the subject matter that we're talking about. Moving on to the actual meat of the lab. So part one, estimating population size. We're going to talk a little bit about the different ways to estimate population size. Like I was saying, can't count every individual most of the time. So these are really useful. And then there's also 2 exercises and then videos and examples in your lab manual for kind of going over these in a little bit more detail so you can kind of get the concept. I remember when I first learned about the mark recapture method, I found it a little bit confusing. So definitely that can be helpful to review that concept. And also feel free to talk to me in office hours if it's still confusing. And then, so there's the mark recapture method and then the transect quadrat method, which will basically be using like a density estimate over a bigger space to estimate the population. And then that is more like what we're doing with the blepharisma today. Okay, so, and then part 2, the hypothesis testing. We're going to talk about what is a hypothesis, prediction methods, and I feel like you've all been learning about the scientific method since you were probably in 1st grade and talking about hypothesis, but I feel like when I first started doing more in-depth science and learning about a hypothesis, it was maybe a little bit different than I first thought, and we're talking about this thing called the null hypothesis, which really confused me for years and years. So we'll talk about that, the difference between that and just a hypothesis, and also kind of thinking through how you can design your experiment to get at the question you're asking, because that's an important part of that too. And then we're going to do breakout rooms, and you will go into your own, you will be talking about your own hypothesis. Maybe, let's see how many people we have, 10. So maybe in 2 or 3 groups, and you can kind of formulate your own hypothesis, and then either I'll pop in to the breakout rooms and talk to you, or we can all come back and kind of discuss it as a group. And this will help you with answering some of the questions on your post lab about, you'll be basically designing an experiment, and we can kind of help refine that and then kind of make sure that when you're thinking through these methods, they're actually getting at the question that you're asking. So, all right, any questions about what we'll be doing for the next little bit? Awesome. Okay, so the first method for estimating population size that I mentioned was the mark recapture. And so we'll kind of do this little thought example. And like I said, there's the video if it's still confusing. But someone in my cohort is doing a mark recapture type study with these, all these very cute rodents. So little guys like this, so adorable. That would be a super fun thing, and you can totally go and do that if you want to be a biologist. Just hang out with teeny mice and gophers and whatever you want. So then you'd probably do some mark recapture. So so suppose you want to estimate the number of field mice in a small field. So one day you capture 100 mice and you mark all of them and then you release them. The next day you capture 150 mice, of which 15 were marked by you yesterday. Calculate an estimate for the population size. So it's basically just a ratio. You've marked 100 and then you only get 15 of those out of the 150 that you've captured on the second day. And so you'll do the 100 that you've marked over the population size, like the whole population size is equal to the 15 recaptures over the 150 that you've captured the second day. And then this will give you an estimate of the total population, which is 1,000 individuals. I know I think that can be like a little bit confusing. And I think that the best thing to do here is kind of think through what the parts are and then where they go in the equation. And then it should be pretty simple. You're like, all right, so I marked 100. That's going to be on the same side as the total population. And then I recapture 15 out of the 150 that I marked the second day. So, and it's just easy cross multiplication then to get the estimate. Does this concept make sense? If it's not, we can go through it again. Also, I recommend just taking some time to sit with it. I actually did a mark recapture study with turtles a few years ago, and to mark them, we would put some nail polish on their shell, and then also you do this thing where you mark their scoots on the outside of the shell, and each turtle had like a unique identifier, like a name basically. And it was really cool because then you got to see kind of do this in action with an estimate of how many turtles there were. And also I got to hang out with a bunch of turtles and, you know, hold them, and they're really cute. So very fun way to sample different populations. Okay, any questions, or we're all thinking through this? That's fine too. Okay, so moving on to the second method, and we can go back if anybody needs to review the other one. But the second one is the quadrat method. So this should be familiar to you because we've talked about quadrats. So using the same technology, basically of just having like a square or circle or whatever shape thing to have a set area of a set size that you know to sample. You'll be basically looking at the quadrats, you'll have them on some grid or randomly placed or whatever, and look at the number of individuals in each quadrat. And then you will take the average of that. And then if you know the area, you can extrapolate to a larger area. So say if each quadrat is 1 square meter and the average of all the quadrats is 3, and you're interested in an area that's like 300 square meters, then you can be like, okay, 3 times 300 is what my estimate for the population over the entire area. Or you can also, you can extrapolate, or you could just get a density. So you could be like, there's an average of 3 blepharisma per square meter. That would be a really, really low density for these tiny creatures. So let's do something like the mice. We can pretend these are mice and say, okay, there was an average of 3 mice per square meter. So this is a really good way of getting at density. Does that make sense? So it can either be extrapolated out, or you can just be looking at like the set area that you're thinking about, and you want to know, this could be good with plants too. For example, you could be like, okay, how many poppies are there per square meter? And then you have these different randomly sampled quadrats, and you take an average, and that gives you an estimate of how many poppies there are per square meter. So imagine doing mark recapture for plants. I think it would be pretty easy actually, because you could just find them again. You'd be like, wow, the plants did not move. So yeah, it's that definitely for different species, you would probably do different methods. I think that for mice, like a quadrat would be kind of hard because they could just run out of the quadrat. So that's why you kind of have these different methods to get at the same question, because, you know, different species have different things that they can do to escape being detected by you. All right, any other questions about the quadrat method or the mark recapture? Okay, awesome. All right, so moving on into part 2, we're just kind of busting right along here, which is great. We're going to be looking at these little blepharisma buddies, and they're pretty cute. They're very, very tiny, and actually it's really cool when you can see them moving around. So in the intro video and data collection video for how to do this. Basically, I think it probably will make the most sense for you guys to watch that on your own time and go through that. But just an introduction to the experiment, these little blepharisma buddies were in different jars with different nutrient levels. So there was a low, a medium, and a high nutrient level jar with a certain number of blepharisma in each jar. And then it was left for 30 days at these different nutrient levels. And we wanted to see kind of how the different levels of nutrients. So basically, think of that as maybe the carrying capacity of the jar, or, you know, what level of resources are available to the population of each jar. It's kind of a unique, or it's an interesting experiment because it's a closed environment. There isn't anything coming in or out besides what we are controlling. So it's controlled, and it lets us really see the kind of pure population dynamics of the blepharisma. So it's a really cool way to think about population dynamics, though doesn't necessarily happen in the real world like this, but that's a lot of scientific experiments also. So for 30 days, it was under these conditions. And then kind of our goal was to see how do these different nutrient levels affect the population? And then something to note is that a high densities blepharisma can be cannibals. So this could be thought of as a kind of density dependence thing, that if there's a lot of blepharisma, then some of them can be huge and eat other ones. So something to think about. And that's pretty metal, I think, for this little guy. Okay, so kind of we'll work through this experiment and an example of what kinds of experimental design you all might want to have in your projects that you're thinking about. You won't actually be doing your own data collection. It's just kind of a thought experiment and a thought process of how you would do it if we were in person or if you had the resources to do it, but still helpful to kind of think through a project all the way through. Okay, so we've introduced the experiment. Now the hypothesis for this would be populations of blepharisma with more food will be more dense than populations with less food. Seems pretty straightforward. Like there's more food available, there will be more resources available for each blepharisma to reproduce. So we believe that the populations with more food will be more dense. So that is the hypothesis. It's just kind of a statement of what you think will occur, a kind of best guess. Does that make sense? Yeah, kind of along the lines of what you've all been taught about hypotheses. So basically just a straightforward, like, I think this is what's going to happen given the setup of this experiment. Then you have the prediction. So this is kind of a more specific outlining of what you want to be testing or how you're going to be trying to get at your hypothesis or answering your hypothesis. So they're talking about the halfway point, basically. 14-day-old high nutrient level population density will be greater than the 14-day-old low nutrient level population density. So you can kind of state it with these mathy terms. You can write it out. So when you're actually writing your hypothesis is the general statement, then the prediction, like I'm going to be looking at this time period and these two groups. And my estimate is that the high nutrient one will be higher than the low nutrient one at this point. So you can use words to describe that. You can just also use these mathy symbols, whatever you want to do. And then kind of, if you do the mathy one, just define what each of the terms is in your statement for your prediction. Okay, so now moving on to the null hypothesis. Does anybody want to take a gander? You might have, I think that this was already introduced in the pre-lab, but does anyone want to quickly throw out what their definition of the null hypothesis is? Okay, it states that the independent variables are not related. So yeah, like basically how I think of the null hypothesis is that this relationship that we think is occurring, we think is controlling the outcome, doesn't seem to have an effect on the outcome. So in this case, the null hypothesis is that the population density will be the same in both populations because we think that the resource levels are what's controlling the density. And if at the different resource levels, there is no difference in the population density, then the null hypothesis is correct, that the population density will be the same in both populations. And this is where you might have, if you've taken a stats class, you might have talked about statistical significance. So this would be that the two populations are significantly different from each other, and you're able to reject the null hypothesis. So in a lot of cases, when the paper or the scientists say that they were able to reject the null hypothesis, it's that their hypothesis that there is some difference among groups was shown to have evidence for it, that it was the two groups were significantly different and they could reject the null. Does that make sense? I know it's kind of a confusing thing, but it's basically, say for, a medicine, a COVID vaccine or something, and you could say the null hypothesis would be that unvaccinated individuals and vaccinated individuals get COVID at the same rates. That would be a null hypothesis. And if you found, like they have found, that the vaccine is very effective and it's statistically significantly better than no protection, then you're able to reject the null and state the vaccine helps. So that's an example in the real world of a null hypothesis. Does anybody have any questions about that concept? Okay, awesome. And then, so yeah, this will be, there's the sampling video and you can collect data in a kind of quadrat method from the video. You'll count the number of blepharisma for 5 drops of the culture and think of the drop as a kind of quadrat that you're sampling from. And then you'll do the average counts, and this is an estimate for population density, so the number of individuals per drop. Yeah, okay, so that is, you'll be able to watch the video and do that for part of your post lab, and then kind of this is what we're doing in the live lab of hypothesis, methods, prediction, and designing an experiment for your own questions. So an example question would be, this is the general question, how does the amount of food influence population size? And then, so this hypothesis that we talked about, populations with more food will be bigger than populations with less food. Prediction, this is like the more specific, kind of zeroing in on what your particular experiment is going to be looking at. 30-day-old population grown under high nutrient levels will be larger than a 30-day one. There's all the little mathy terms. And then a good way to think about if your prediction and hypothesis are related to your question is think about what a graph of your data would look like. Like, how would you show what you're trying to show, and making sure that the data that you're collecting is really answering that question. And then the methods, how would you conduct your experiment in a way that someone else could repeat your methods? So describing, just like how they had the experiment that we saw with the blepharisma, you could say, I would have 3 vials of each of low, medium, and high nutrients. I would measure the populations every day for 30 days. And then at the end, I would see, or after 14 days, after 30 days, I would see which populations were bigger, etc. So that is kind of a way of describing your methods. And then in this one, they've even more specifically said, like, oh, I would take 5 drops of each and use the quadrat sampling method to evaluate the density of blepharisma. So how would you evaluate the population also? Because you probably can't count every blepharisma in the entire jar because there's so many and there's so little. So you're going to want to think about how you would sample for that. So I think in the normal lab, this would just be kind of limited to the blepharisma concept, since we're not really going to be carrying this out. Pat said it was fine if you wanted to think about another species that you might want to sample for. That might be a little more challenging because we haven't kind of gone over the best way to do it for different species, but we did go over when each sampling method is better, so you can think about that also. So yeah, feel free to choose another species to think about and just kind of think about a general question that you want to know about the population. You could think about 2 different populations and 2 different resource types of the same species and thinking about, is one better for density or something like that. But then you're also going to want to think about how to control the experiment if you can. So let's see, I think we might have lost some folks, but that's okay. I think I'm gonna split. Are there any questions about what we'll be talking about? So just kind of setting up your whole hypothesis, methods, predictions for an experiment, chatting about it with your group, and then I can pop in and talk about it with you, make sure that we're all on the same page, and then hopefully you'll be in a good place to complete the post lab. I'll probably just give people like 5, 7, let's say 7 minutes to talk about it. And yeah, so I'll give maybe people 5 or 6 minutes, and then I can pop in and just wait for me to pop in. And let me do the breakout rooms. Any questions in the meantime? Okay, great. All right, so you're going to, I think you're just going to go out there, but I'll see you in a few minutes. Good luck.